right. Our next presenter is going to be someone that you've heard from almost as much as me. Laurel at a previous meeting won her K-Bump jacket for being, I think at the time, a 12-time presenter, and it's more than that at this point. So let me just get everything going here. And being the dedicated person she is, she came off the river to come talk to us again.
It's a good trip when you get to be on the river and present at K-Bump.
All right. Wait, let's go. No, all right, take it away, Laurel.
Thanks. Thanks everyone for hanging in there with lunch quickly getting close here. And I'm going to be presenting here on the Quartz Valley Indian Reservation special study on attached cyanobacteria and anatoxins throughout the Scott River Watershed. I presented on this 2 years ago when they were 2 years into their study, and they've now completed 4 years of data collection. This is a really cool project. There are very few data sets out there anywhere that have this much information about these attached mats that produce anatoxins. So it's becoming a really impressive data set, and we're learning a lot about the Quartz Valley, I guess the bigger Scott River Valley from it, but I think there's a lot that others beyond just the Scott watershed can also learn from the work that they're doing. Anatoxin is a growing concern in freshwater ecosystems. It's been the cause of dog deaths throughout the West and other places around the world. We're seeing newspaper articles becoming more and more frequent about dogs dying after going to the river and consuming algal mat material. It was the cause of death to a huge group of flamingos in Kenya a couple of decades ago, which was one of the first sort of sentinels that this is becoming an issue and something we need to learn more about. Anatoxin is a potent neurotoxin. It has the nickname very fast death factor because if consumed in high quantities, it can actually cause a really quick degradation and cause respiratory failure that leads to death if consumed in high quantities. So it can be a scary toxin, but what we know about it is largely based on these planktonic blooms. So we see a lake or a reservoir, we see it turn green, we know there's an algae bloom. We know a whole lot less about the algal mats that are attached to the bottom of the river. They're way less visible. We're not looking for them. They don't have that same gross green look to them. This graph is just showing publications about algal blooms, and most of those publications have been on these planktonic blooms, not on these attached or benthic blooms. And so we just know a whole lot less about the stuff that's growing on the bottom of rivers compared to these lakes and reservoirs. That being said, the detections of these attached anatoxins or mats that produce anatoxins, it is increasing globally, but you can see that it's just been in the last decade or so with this lighter green and then kind of cyan color that countries throughout the world are starting to detect mats with anatoxin in higher quantities. And it is kind of interesting. You see Kenya there, and that's because of that flamingo incident a couple of decades ago. But we're starting to pay attention. We're starting to find it more places where we do look for this. We're finding that it might actually be pretty widespread. New Zealand has been the leader in this research. They have done a ton of sampling throughout both the North and South Island. They've found hundreds of rivers that have had these cyanobacterial proliferations. Most of those proliferations have had anatoxins in them. A little bit closer to home, the Eel River has also had quite a bit of work done on it, both by researchers at UC Berkeley as well as the Water Board. And they're also finding widespread mats of cyanobacteria and a lot of anatoxin in those mats. And so on the Klamath River, there had been a lot of sampling for cyanotoxins over the years, mostly because we had this huge microcystin problem in the reservoirs. So some of those samples that were tested for microcystin were also sampled for anatoxin. When we came through and, oh boy, okay. When we went through and analyzed those samples that had anatoxins, we saw about 6% of those samples were had detectable levels of anatoxin in them. So we started to ask, like, hey, where is this anatoxin coming from? Is it coming from the same source as the microcystin? And a 2016 study looked at aniseed genes in those in 126 water samples. So they found even a higher concentration of anatoxins. So we know it's out there. We know it's in the water. Where is it coming from? Well, in 2019, we started looking and finding some of these attached mats. And when we sampled those, we found a much higher rate of anatoxin detection. And that led us to ask, to what extent are these attached cyanobacteria producing anatoxin in the Klamath River? Is there a source that's different from the reservoirs that is the cause of the anatoxins? So I worked really closely with the Karuk tribe in 2021, and we did a study throughout the Klamath River and some of the bigger tributaries to look at how anatoxins were distributed throughout the basin and how they varied among sites and through time. And then depending on if we sampled the water versus the mats, we looked at the toxins, who was producing the toxins and what conditions may have promoted them. That's available in an open source publication if you want to know more about that 2021 study. The big take home there was that we found cyanobacteria pretty much all over the place when we looked for it. We found these attached mats on the margins of the rivers. When we tested them for anatoxins, they were quite common, especially later in the summer in August and September. Those filled in dots show the sites that we sampled and where we found anatoxins. You can see in July, we sampled at sites with fewer anatoxins, but really common, especially late in the summer. And you can also see that the Scott River sites had some of the highest concentrations of anatoxin. And so a lot of folks from Quartz Valley Indian Reservation, including Sarah and Crystal, who are both in this room, got this project started and have kept it going. There's a lot of good folks that have been out in the field doing all this data collection as well over the last 4 years. And we wanted to take what we learned from that Klamath study and see what we could learn about those high samples that we found in the Scott River. So here, we asked to what extent anatoxin from attached cyanobacteria is a concern in the Scott River watershed. And some of our questions could be grouped into these different categories. First, we wanted to think about field and lab methods and how we can really efficiently and effectively monitor for anatoxins in the Scott River and tributary streams. And then to look at temporal and spatial patterns, like how it's varying seasonally among years and what that spatial extent of anatoxin production from these attached mats are. And then we're also just thinking about like what taxa are out there, who's actually producing the toxin, are there other cyanotoxins that are an issue? So these are all things that we're thinking about when we set up this study and when folks have been out testing. There's been a whole lot of samples over the years, both in the mainstem Scott River sites and the tributary sites throughout the Scott River watershed. And you can see there's been around, 6 to 8 sampling efforts throughout each season at most of these sites. Sometimes we did a little bit more of a shotgun approach to expand the different sites just to get a bigger spatial picture, but don't need to go through this, just want to show you that there is a lot of sampling that's been happening between 2022 and 2025, both seasonally and across space here. Okay, so jumping into some of the results, spat samplers are a really cool way to measure, or I should say, to sample cyanotoxins. They sample the dissolved toxins in the water. And what's so cool about them is they're really easy to deploy and collect, and you don't need any sort of ID skills. You can just put this in the water, set it out there for a week, collect it, send it to the lab, and you can get a semi-quantitative measure of the dissolved toxins in the water. So the Quartz Valley has deployed these throughout 3 of the years of sampling, and in partnership with the Water Board here in 2025, they were able to deploy a bunch more of them. And what you can see in this plot on the left is that we do detect anatoxins regularly when these spat samplers go out. The dotted line here is the detection level. So anything below that dotted line was a non-detect. We didn't get any anatoxins. But these samples above the dotted line did detect anatoxins. And so you can see that the Scott River site specifically, which is the kind of green and yeah, I guess both green tones there, frequently had anatoxin detections. The purple site in 23 and 25 had some anatoxin detections, but less. That was at Kidder Creek. But we are finding that these spat samplers seem to be less reliable than surveys of benthic mat samples. And so in the Quartz Valley Indian Reservation Environmental Department, there's folks that are out there regularly. They know what these mats look like. They're really good at getting in the water, looking for these mats and finding the target algae. And because of that, those trained field technicians and biologists are able to find what they're looking for and actually get a much higher detection rate than the spats. And so what these two plots show are the Anatoxin genes, which is one of the laboratory measures, and the ELISA measurements on the far side there of the plot. And you can see that there's quite a few samples under that dotted horizontal line. And those are spots where the spats did not detect anything, but we did have detections from the mats. So all to say, these are a really awesome tool, especially if you're not able to field ID them, or if you're not able to get in the water and really search as much, but we still are having a better detection rate with the direct mat samples. We trialed drift nets in 2022, and you can see this is just an invertebrate drift net, but we used it to capture floating algae moving downstream. And we did find that it was a useful way to capture this material. And you can see on these plots that what we call these CPOM samples or algal mat genes on the x-axis, they that's on the y-axis. So they corresponded well though to just the mats that we sampled. We did find that this isn't as useful in small streams because you're catching a lot of leaves and sticks. So this is a cool option to have if you're sampling in a larger river, but it does take more time, it's more equipment, and overall field technicians found it a little bit more cumbersome. So we dropped this in the Scott, but if you're sampling in a bigger river, this could be useful. So transitioning to thinking about lab methods here, we sent a lot of samples in to be analyzed, both using the qPCR technique, which specifically targets the Anatoxin gene cluster, which is a requirement for anatoxin production. And then we also compared those to total anatoxins by ELISA. And we get this really nice correlation between these two different methods. And so here we're finding that these qPCR laboratory methods, which are more affordable than the ELISA tests, seem to be a really good indicator of toxin production still. And so if there's a budgetary constraint, that might be a good option to think about for organizations who are sending in samples and can't afford the ELISA method. It's not as direct of a toxin measure, but given the high amount of variability in benthic algae and riverbeds in general, it does a pretty good job. So we're excited that that worked as well as it did. There were a few samples here where we had low amounts that were non-detects, but those were all and that really low rate that weren't showing much of a public health concern. Okay, so what did we see in the Scott River Watershed across space and time? Well, spatially, anatoxins were pretty rare in these smaller tributaries. We did find extensive cyanobacterial mat coverage in these tributaries, including Etna Creek, Sugar Creek, Shackelford Creek even. There was a lot of cyanobacterial mat coverage, but the only tributary with consistent anatoxin detections have been Kidder Creek. And those have been pretty high at times. Otherwise, the other sites where we're having high anatoxin detections are all in the main stem of the Scott River. And those are really most concentrated at these more downstream sites, so below where the Shack Mill confluence comes into the river. And unfortunately, at some of these really high public use sites like Scott River at Jones Beach and Indian Scotty are having the highest toxin concentrations found. And I'll just say some of these are very high. They're mat material, so you'd have to have like a condensed sample to do harm, but they are very high rates of detection and high concentrations at those sites at times. Okay, so across years, we're seeing a consistent seasonal trend. And that seasonal trend is that the highest anatoxins are occurring in late summer and really even into autumn and all the way up until the first flush is happening in the fall. So you can, there's a lot of variation here, but you can definitely see this increasing seasonal trend. The earliest sampling has ever even occurred is July, and that's kind of the earliest any mats are being seen. And so it seems like it's really waiting until the flows come down, those mats start to grow, and they continue to grow and continue to produce toxins into the fall. And kind of interesting here, Eli took one sample in 2024 all the way in mid-November, and that was the highest sample for the whole season. Okay, so here, because we've had a lot of variation in which sites were sampled in which years, I just want to show you all differences among years, just comparing those two sites where we had consistent data collection at the same time. And those sites were Kidder Creek and then the Scott River between Jones Beach and Indian Scotty. And what you can see that in general, we had higher concentrations of anatoxins in 22 and 23 and kind of lower concentrations in 24 and 25. With the exception that Kidder Creek was higher in 25, I'll just throw out, it'd be really interesting to know more about what the fire there had done and if we were getting more nutrients. We definitely aren't getting into that in this kind of more like general patterns discussion, but a lot of cool questions to think about there. I do want to throw up some context with the hydrograph when we're thinking about these 4 years. These numbers here are the average anatoxin concentration at Jones Beach for each of these 4 sampling years. And you can see very low flood year in 22, and you can kind of see that increase in winter flows here from 22 through 25 and a decrease in anatoxins. This is an NO4. You can't say anything statistically, but it is really interesting to start looking at these patterns and considering how flow might be influencing the toxin production throughout the Scott River. Also, you don't really see the differences in summer flow, so I just wanted to plot those there. That's just the mean flow from August to September. And you can see that there was also much substantially higher summer base flows in these 2 recent years where the anatoxins were lower. So definitely something to think about as folks start developing more research and trying to think about the drivers of these toxins. Flow or something that is correlated with flow may very likely have an influence in these higher toxin production times. Okay, in terms of what taxa were actually out there, I'm going to direct you to the Klamath study first, where we found quite a diversity of cyanobacterial taxa in these mats. But you can see in yellow that microcoleus was quite common throughout the Klamath. We also saw a lot of anabina in that study and various other taxa, including microcystis, which were probably coming down from the reservoirs and getting entrained in some of those benthic mats. So more diversity, still quite a bit of of microcoleus. If we look at the Scott River over here on the left, it is definitely dominated by microcoleus. We're seeing microcoleus as the main taxa, which is good because those microcoleus mats tend to be what we can visually target. But more and more, we're learning that from the bigger research community that microcoleus is the likely anatoxin producer in these mats. So even though there's like a lot of mix of different taxa in the mats, we're learning that microcoleus is probably the one producing the taxa. This is a great resource for anyone that's interested in learning more. This paper just came out in January this year, the global proliferation of aquatic benthic microcoleus. It talks about toxin production, ecology, kind of a synthesis of what we know right now about the microcoleus, which was the dominant taxa in the Scott and likely the producer of anatoxins there. We did look at other cyanotoxins and consider that other toxins could be an issue, but as you can see here, this is just an average of all the samples that we had processed for other cyanotoxins, and that anatoxin in those subsample that we ran with all toxins was almost always detected, with the exception of just a couple of samples, with a mean of 100 or over 1,200 micrograms per liter. And these other toxins had relatively very low detection. So they are out there, but, and I threw these on here, these are some EPA drinking water limits. They are generally at levels below even drinking water limits. And I just want to put the caveat on here. We're talking about mat sample. These are drinking water limits, thinking about like a liter of water. So when I'm throwing on these numbers, you have to think about if you were to consume a liter of mat slurry, right? So like that would be a lot of gross algal material to consume. So when you take that into context, these other toxins are occurring at very, very low environmental conditions. And I think like anything, you can think of toxicity as being dose dependent. I see these numbers, I'm not super worried about them, with the exception of anatoxin, which you can see is orders of magnitude higher compared to these other ones. Okay, so I think bringing in these next couple years of data into what we had already looked at, something I had presented at KBMP a few years ago, we're really confirming the findings that we initially found in the 2021 study in the Klamath and the first 2 years of the Quartz Valley study. And here are some recommendations that come from that. I think first and foremost, continuing to focus these visual surveys on these microcoleus mats, given that that's the most likely toxin producer in the system, is a good practice. If you have the capacity to prioritize mat samples, that's probably the most effective at detecting anatoxins and knowing if there's a risk. That's also what a dog is likely to lap up, right? That's the most kind of direct contact. Spats are also a super useful tool though, if you don't have ID skills or need to just get something in the water to have some sort of consistent measure. qPCR is more affordable and I think presenting to be a really good indicator of toxins. So that's a great option to have. And I think overall, thinking about where are we looking for benthic cyanobacteria, it can be in really diverse habitats. It can be in these higher water quality streams, including streams like Kidder Creek that are coming out of the marbles. And so keeping our eyes out even in water that we think of as traditionally really high water quality streams is important when we're thinking about potential anatoxin risk. And then if you have to choose a time to sample, really focusing on late summer and fall is continually what we're finding in terms of the highest potential toxins. Where do we go from here? We think that benthic anatoxins are probably a lot more common than previously expected. And so it is something to be aware of, not necessarily scared of. It might very much be that these mats are a little bit like wild mushrooms in the forest where there are toxins out there and we just need to learn how to avoid it and make sure we're not consuming it. And so I think public awareness is going to be a big part of this and trying to make sure people are aware but not scared. We need a better idea, though, and understanding of what promotes microcoleus, and if it is a growing problem. We have not done enough research to know to what extent things like flow or nutrient pollution might be promoting either the extent of mat coverage or increased toxin production. We had QVIR had a partnership with a student from the University of Nevada, Reno, doing a study from 2024, trying to understand some of the drivers. Unfortunately, that year had really low toxin production, so she's not getting the variation she needs to really show what the drivers are. Hopefully, we'll still learn some cool stuff from that, but it's a hard thing to study because we don't know when we're going to have a big toxin year. And again, just making sure that we're educating folks, making sure they know that you need to keep your eyes on dogs and kids and make sure they know not to put any kind of algal material in their mouth. And I just want to plug this really cool resource. This is another student from that same lab did some work on the Salmon River, and she produced this really cool Salmon River coloring book that has a bunch of pages with algae cells and then bigger river pages. And there's some really cool information about algae and ecology and identification in this coloring book. And it's available online. If you Google Salmon River Coloring Book, you can access it. You can download it, print it, share it, use it as much as you'd like. Jordan did a really awesome job. It's all hand-drawn and a really fun resource. So please make use of that and let us know if you are able to use it so that I can pass it on to her that people are enjoying it. And with that, I'm happy to take any questions if there's time, or we can talk during lunch if you're ready for that.
Any important questions that won't get in the way of sandwich time?
Many people in the Klamath Mountains, including myself, get their drinking water from surface streams, often unfiltered. Some of these smaller tributaries have had increased fire, increased algae after fire. Is there any danger in places where maybe these algal mats are producing toxins in consuming does the toxin end up in the water column if you're consuming water that's coming from a creek with these algal mats?
Yeah, the biggest risk is in the mat material. Anatoxin breaks down really fast in UV light. And so you really want to make sure that you don't have chunks of anything in your water, because that's where the risk is going to be. The dog deaths that have been associated with anatoxin, when they've biopsied them, they've had mat material like filling their stomach. So this is a situation where it seems like you need a relatively high dose, and there are no recorded deaths to my knowledge of a human death just because you'd have to eat quite a bit of it. That being said, I would look for chunks for sure and make sure if there's like floating mats to really avoid getting any of that in your water source. The dissolved toxins tend to be less of a, they're not very common in these flowing clear waters. It's in that algal mat material.
Knowing that the regional board uses the ELISA results to determine public health postings, has there been any discussion with them yet about the qPCR as a methodology and setting, thresholds that can be kind of equivalent?
Yeah, we've talked about it, not in a formal context of establishing them, but I think that we have enough data that we could easily, I put regression lines in it. We can do an easy linear regression and kind of find out. But their methods too for posting are a little bit more qualitative than that, because you have to consider the extent of mat coverage, how high are the toxins. This is a really hard thing to come up with a public health threshold for. And so my guess is that they're going to say like, sure, this is one more good line of evidence. But I think what they're looking for is like, do we have multiple lines of evidence from mat coverage, qPCR, or ELISA? Like, what are we seeing?
Yeah. And a ballpark cost difference between ELISA and the qPCR?
Oh man, last year I think the qPCR were like $90 a sample and the ELISA were like $160 maybe. And if you filter it yourself, then it might be like 60 versus 160. So it could add up.
There's a question online from Desiree Tulos. Would you feel confident snorkel sampling in a river with wide coverage of microcoleus?
I have and I do occasionally snorkel in a river with broad coverage of these mats. Yeah, I feel comfortable because I'm pretty good at keeping my mouth closed. Like I would feel less comfortable with a 4-year-old doing it. Yeah.
Great. All right. Any other questions? Yes.
I was just wondering, with microcystin, there's like real clear management ways you can deal with it. Do you know is it just like reduced nutrient loading or obviously we can't control if there's flushing flows in the Scott, but is it like flushing flows and reduced nutrients? Any thoughts?
We don't know. We don't know anything. I shouldn't say that. People just really started documenting this within the last decade or so. And it has provided super challenging to relate the environmental drivers of mat coverage and toxin production, in part because it's really hard to sample. So until we are really good at saying like, what's the variation from month to month and year to year and river to river, we can't really connect that variation very well to the drivers. That being said, there's a lot of lab work being done to try to like manipulate it in the lab and see how it responds to different nutrient levels. But then sometimes you go out in the field and things don't work in a real river like they work in the lab. So unfortunately, we're very much still in this like learning phase, and we need more research to try to understand to the extent that it is flows or nutrients or some combination of flushing flows and nutrients. And I think until then, that's when kind of public education and just making sure that, the vulnerable population, which kids, dogs, wildlife, I guess we can't do much about that, but just making sure that we're educating folks and trying to be cautious of our interactions with it.
I'll just say very briefly, Kodak Tribe has been collaborating with the Water Board on early detection methods for anatoxin mats, and we use spat samplers and visual surveys, which brings us to a threshold level. And then if that happens, they do more testing, and I do think that reduces the cost somewhat, but it is a non-quantitative measure that was developed on the eel, right? A spat sampler is not going to give you a volumetric measurement. Or in the Klamath it won't, but
which unfortunately though we're finding in the Scott, and it might be different in bigger rivers, water volume is a huge potential complicator. In the Quartz Valley study, we're finding that if you see a mat, that's going to have a detection before a spat does. So the spats in some case might actually be like a late detection once the toxins are high enough to start accumulating in that spat. So if you can sample the mat first, it seems like that might be the most like just preemptive way to identify it.
Absolutely. Yeah. And I think the spats, it's an unknown distance upstream that they might be capturing that material from. So,
yeah.
Great. Any other questions? Sounds like applause was really where we are. Well, thank you so much, Laurel. And thanks to all the speakers from this morning. Let's go have some lunch, and we will start up again at 1:10. So a little less than an hour, but we can work with that. All right, thanks everyone.